Hello everyone, I Anish. I welcome you all on the behalf of A.S.S Foundation. As you all know, we are starting our new batch from July 11. So those who have not registered till now, please do it ASAP. So now we are starting our new lecture on the topic cells of the immune system. This slide here represents the overview of this lecture. We will be starting with the hematopoiesis, like how hematopoietic stem cells is divided into the lymphoid lineage, which further makes the natural killer cells, B cells, and T cells, which are also known as lymphocytes. Then we will be discussing the myeloid lineage, which further makes the neutrophils, eosinophils, basophils, mast cells, and the dendritic cells. And finally, the erythroid lineage, which will be discussed in the human physiology part, how like RBCs are made. Here, IL represents the interleukin, which are the cytokines, basically the chemically, these are proteins, small molecular weight proteins that help with cell signaling and the cell differentiation process. So here in this lecture, we will be discussing the hematopoiesis part, like the formation of blood cells and its components, the lymphoid and the myeloid lineage, and the types of leukocytes, and the last but not the least, what are primary and the secondary lymphoid organs. Okay, here the only the lymphoid lineage is mentioned in this box. Like common lymphoid progenitor makes B lymphocytes, T lymphocytes, and the natural killer cells, which further go to the lymph nodes where they they keep on maturation stage, and after they are released, like B cells will form plasma cells, T cells on uh, combination with antigens or the activation leads to the formation of activated T cells, and further natural killer cells will be acted to the activated natural killer cells. And in the myeloid lineage, we will be discussing the functions of neutrophil, eosinophil, basophils, mast cells, and the macrophages. So let us begin with the types of leukocytes. Leukocytes can be granulocytes, like which contain granules inside them. And what exactly is the function of granules? Why there are granules in one type of leukocyte and is absent in another type of leukocyte? The granules basically contains cationic proteins, proteolytic enzymes, lysozyme, and myeloperoxidase, collagenase, and acyltransferases. Like as these names or enzymes suggest, these helps in the degradation, like degradation of foreign particles or the microbes or the pathogens. And in granulocytes part, we will be discussing neutrophils, basophils, eosinophils. And in the agranulocyte part, we will be looking into the monocytes part. Here, on centrifugation of blood, in the upper region, we will be looking into the plasma, which contains 92% of water by weight, and it contains 7% proteins, out of which albumin has the highest composition, and it also contains some of the electrolytes, nutrients, and the respiratory gases too. And the middle layer consists of the buffy coat, which consists of platelets and the leukocytes, and the last part, that is red, dark red in color, contains erythrocytes or the RBCs. So now one by one we will be discussing the granulocyte part. See, in the slide, granulocytes will look like these. We will here looking into the neutrophils. Neutrophils, these can be stained by both acidic as well as basic dyes. Here we can see the nucleus of neutrophils are multi-lobed and the lifespan of neutrophils are of few days. The function of neutrophil is that is it is involved in the phagocytosis and the activation of bactericidal mechanisms. Phagocytosis can be of with the help of reactive oxygen species or like direct engulfing of the microbes. In the next part, we will be looking into the basophils. Basophils, as the name suggests, they are stained with the basic dye that is methylene blue. It contains lobed nucleus. They are less than 1% of total WBCs and they have high affinity receptors for IgE antibody. They are involved in allergic reactions. The next one is eosinophils. They have bi-lobed nuclei and they can be stained with acidic dye. They are granulocytes that are present in blood and contains the enzyme that are required to damage the cell wall of parasite. The granules of eosinophils contain basic proteins that binds to the acidic dye. They also release reactive oxygen species and cationic peptides that attack protozoas and helminths. And in the next, we will be discussing the agranulocyte part. In agranulocytes, there are monocytes. These monocytes are mononuclear, and when they enter tissues, they can form either macrophages or dendritic cells. Macrophages helps in the phagocytosis and in the antigen presentation too, while dendritic cells are involved in antigen uptake in peripheral sites and also involved in antigen presentation. When we talk about the myeloid lineage, which we have discussed till now, these cells are involved in the innate immunity. But while we talk about the lymphoid lineage, these cells are involved in the adaptive immunity. These consist of lymphocytes, 20 to 40% of RBCs, and they are present in lymph and lymphoid organs. Lymphocytes involve B lymphocytes, T lymphocytes, and natural killer cells. Now let us start with B lymphocytes. B lymphocytes expresses membrane-bound antibodies. They can differentiate into plasma and mammary cells. They serve as antigen-presenting cells. Further, we will be looking into the maturation of B lymphocytes. The next one is T lymphocytes. These are involved in the production of cellular immune responses. Here, T is derived from thymus, which is the site of its maturation. And there are two types of lymphocytes that are Th lymphocytes, which means T helper cells. These are represented by CD4 glycoproteins, which are needed for defense against the intracellular and extracellular pathogens. They stimulate B cells to make antibodies. The next one is cytotoxic T cells, which are represented by CD8 plus glycoprotein. These are needed for defense against the intracellular pathogens. The next one is natural killer cells. They are like TC cells which are involved in killing, but they destroy intracellular pathogens not by phagocytosis but by perforin and granzymes. In this, there are no antigens needed and there will be no memory as like TC cells. There are two special type of effector and memory lymphocytes that circulate through the normal bloodstream and are responsible for systemic immunity against a particular pathogen. The memory of cells are important for providing protection against second exposure of antigens. They are produced in body during their first encounter with the antigen and expand following the repeated exposure. Now we will be looking into the lymphoid organs. There are primary and the secondary lymphoid organs. Primary lymphoid organs involve the maturation of T and B lymphocytes. In thymus, maturation of T cells occur, and in bone marrow, maturation of B cells occur. And bone marrow is the major site for the generation of circulating RBCs, granulocytes, monocytes, and B cells. Majority of B cell maturation take place in bone marrow, but the final maturation completes in the secondary lymphoid organ, that is spleen. T cell maturation occurs entirely in thymus, while natural killer cells maturation occurs entirely in the bone marrow. The common myeloid progenitors give rise to lineages of erythroid, granulocytic, megakaryocytic, and monocytic cells, which give rise to RBC, granulocytes, platelets, and monocytes. The secondary lymphoid organs involve spleen, lymph nodes, and malt. Malt is mucosal associated lymphoid tissues. Skin, gastrointestinal mucosa, and respiratory epithelium mucosa have their own lymph nodes, and they help in trapping the foreign substances and the antibody production. So that's all for the today's lecture. In the next lecture, we will be discussing the antigen part. Thank you.